Thank you very much for the introduction and the opportunity for speaking here today. I have the privilege of being the last talk before the social as well, so I'll try to be a little bit quicker as well. So with that, I start off with a little bit of a morbid slide, I guess. So there have been, over the last couple of decades, many different infectious disease outbreaks. These are just a few. I have missed many others that are more regional to other parts of the world as well. And really what this was showing is that there would be one potentially that leads on to what would become a worldwide event, this sort of next pandemic, and that ended up being sort of SARS-CoV-2, I guess, in recent times. So what have we kind of learned from this? What can we do to take things forward? And this is what my talk mainly will be kind of covering here. So looking to develop the infrastructure to deal with that next pathogen through resources and databases at EMBL-EBI and other project collaborations, for example. So just some context. So a few of you, many of you are probably aware of this, but the European COVID-19 data platform was launched in April 2020 as a response to the outbreak for SARS-CoV-2, which was declared a pandemic in March. And here, what it really aimed to do was support and enhance access to COVID-19 and COVID-19 related biodata. It sits on top of existing infrastructure at EMBL-EBI, so it taps into all the individual sort of resources, different data types at those resources, and so on. And it includes three main components. So you have the COVID-19 data portal, which is the interface, the SARS-CoV-2 data hubs, which are essentially a set of tools that support submission, analysis, visualization, and presentation of COVID-19 sequence data and its analyzed outputs. So we've been working sort of with the public data in the portal through these SARS-CoV-2 data hubs, but this was really with the aim of enabling certain tools to become available for pre-publication or private data sets, for example, with an embargo before they're released. Which was the initial idea of the data hubs that were based on the Compare data hubs. The final component is this federated European Genome-Phenome Archive. So this is all about sort of the human COVID-19 biomolecular and phenotypic data, and it's hosted by the European Genome-Phenome Archive at EMBL-EBI, along with collaborators as well. And the platform is supported by various projects at the top, and going forward recently by COVID, Recoded, and VO have been quite important going forward as well. So just a few highlights here. These are very big components, and to summarize them into one bullet point is not easy. So the portal supports more than 25 million records across all sorts of different omics and also other non-omics data types. Some of them are hosted directly at EBI. Some of them are beyond EMBL-EBI as well through collaborations. And there are a network of 12 national data portals that support the central sort of portal. And that provides more regional information, access to other data and databases, etc. The data hubs, I mentioned the public sort of data analysis. Through the mobilization of more than 6 million raw read data sets, we've kind of systematically analyzed more than 4 million of them, which provides a very big data set. And has supported these public health reports that combines the genomic sequence data along with sort of circulating variants of concern, variants of interest at any given point in time. And the human data side here, so Zara presented her poster here, and this is kind of what this is mentioning here, where this linking, the ability to link between different types of data within sort of the cohort data set level. I didn't mention this as a specific component, but this is definitely one of the key components, the training and outreach side. So here we've been lucky enough to participate in these sort of online and in-person tutorials and workshops that are supporting researchers across different networks. We've also documented and provide online videos and so on to cover submissions and usage of the platform. And we're kind of trying to generalize this and go beyond just COVID-19 to this sort of general pathogen submission guide with a dedicated sort of help desk email, which will go directly to a member of our team. And the last one there, just working with the EMBL-EBI training team recently, who have collaborations with the Cabana project, for example, in Latin American researchers. So tapping into the network and supporting their needs in terms of for SARS-CoV-2 as well. In terms of usage, this is just to highlight that the platform is really part of a really big ecosystem of different things, and this is only showing examples of things. So on the left, you have an example of another downstream resource, such as open targets. The small bubbles on the left kind of present just how many different types of data are being pulled into the resource. Some of those are coming from the portal, for example, and that enables for this systematic discovery of drug targets, for example. You have very much these cloud workflows. That the Galaxy project is an example, but there's lots of other examples of other groups who will be either doing analysis on the cloud or their own HPC clusters and so on, or high-performance clusters and so on. And then there's a lot of other sort of downstream visualizations that add more context to the data that's in the portal. So things like CRG viral beacon, next strain, COV spectrum is another one, but there's, again, many of these that exist as well. And I mentioned the other two on the bottom in sort of the initial slides in terms of the summary of the portals. So with that, what do we kind of have? What can we take forward to a more general approach, things to focus towards priority pathogens, for example? So what we are fortunate to have is this sustainable model. So being part of the European Nucleotide Archive at EMBL-EBI, EMBL-EBI itself is a large source of public biomolecular data. As you can see, it kind of covers many different types of omics and other types of data. I think the ENA falls in the second column there. But overall, it has the vision to benefit humankind by advancing scientific discovery and impact through bioinformatics. And so with that foundation, we kind of build these molecular biology components are built on top of them. And that means that you have these resources with the institutional commitment and history of sustained activity. And they are typically connected globally through data exchange collaboration. So for example, as many of you know, the ENA is part of the International Nucleotide Sequence Database collaboration with partners in NCBI in the US and DDBJ in Japan. With this comes open standards as well as software standards around data or metadata. I use the word, again, loosely, I guess, in this context. But these resources will collaborate with the communities and other consortia, and what this means is you have this very high level of distribution of expertise and effort, so the appropriate people are around the appropriate types of data there. And I also mentioned the Global Biodata Coalition, so this is Guy Cochran, who leads our team, has also got a role in GBC, and they're trying to map the landscape of sort of data resources that need to be sustained for long periods of time. So with that sustainability, with what we've learned for the COVID platform, we kind of officially launched the Pathogens platform in end of June this year. It did exist before under the Compare project initially, but officially launched with the sort of COVID-19 data portal framework going forward as well. So the scope here is quite broad. It's all pathogens, all disease, focusing on the host, vector, and pathogens, but also certain themes of activity such as AMR. And through and one of the angles of this is, again, preparedness and outbreaks. And so what we've done as part of the platform is essentially roll out the pathogens portal and the pathogen data hubs officially as well. So the portal is the interface of all the pathogen data or biodata in EMBL-EBI, and we're hoping to kind of expand that as we go along as well. And then the pathogen data hubs are essentially what I mentioned for the SARS-CoV-2 data hubs, but provide this sort of infrastructure to be in the state of readiness for when an outbreak arises, so being able to tap into tools and so on. And monkeypox provided this example or to try and apply some of the tools that we've learned in a different scope. And so we managed to do that for example, some of the submission tools, like the drag and drop tool, analysis pipelines, submission instructions, and so on. And that data is now available through the outbreaks page, which has changed since the launch as well. Specifically on outbreaks and preparedness, this is essentially showing a dedicated page for open data on the World Health Organization's priority pathogens, along with other pathogens from data coordination projects. So we're trying to cover here bacteria, viruses, and eukaryotes, namely fungi from that list. And essentially what this page is presenting is data holdings, ability to advance search across the different types of data, and then at the moment, next strain reports is just an example of reusing open tools and infrastructure and kind of embedding that, for example. So we're really grateful for the next strain team for the work that they've done there. So aim is to cluster related data and tools. And one of the interesting things about this page is this organism facet on the left-hand side presents the number of data sets per organism. And so what you'll be able to see is where the gaps lie in research, for example, or in terms of data sharing, maybe. So it's quite a neat page to look at. Lastly, I just want to highlight this Pathogens Portal Users Forum. So this is for those who might be interested, or a researcher or data scientists or people within the groups that might be interested in pathogen data. And we have a feedback session on the portal on the 14th of November. It is a hybrid event. Unfortunately, we aren't able to pay for travel and accommodation, but during the time the things will be covered. So there's a QR code and the form that's linked there. And with that, huge thanks to everybody that's been involved, many different collaborators across different projects, communities, and of course partners. So huge thanks to everyone, and thank you all for listening as well.